Hello students, good morning to one and all of you. How you all are doing? So I believe that you all are absolutely right and doing excellent in your studies. So students, I again welcome you to another class of 9th Science Biology. Lesson number 5, that is a fundamental unit of life called cell, right? So students, till your last lecture, you have discussed about, that is, nucleus. In last lecture, you discussed about nucleus, and there you learned about the structure of nucleus. There you also discussed about the important functions of nucleus. So in the structure of nucleus, you have discussed that nucleus, it is a double membrane bound organelle which is present inside a cell, and the two membranes, the outer membrane is rough and the inner membrane is smoother. Outer membrane is rough because it is having attached ribosomes, right? Then you have discussed about the internal structure of a nucleus, that inside our nucleus it is containing the floor of the nucleus. What I mean to say is made up of a jelly-like substance which is known as nucleoplasm. And inside nucleoplasm, you see entangled mass of thread-like structures, and this is known as the chromatin material, right? The same chromatin material which is made up of DNA and proteins, and same material condenses down and form rod-like structures which are known as chromosomes. And I hope you still remember that chromosomes inside a cell are only visible when a cell is undergoing cell division, right? And then you have studied about the function of nucleus. So the main important function is that nucleus is referred as the master of the cell. Means whatever activities are going on inside a cell, these activities are controlled, regulated and maintained by the nucleus. Say for example, for our body, the master is the brain, right? In the similar manner, for a cell, the master is its nucleus, right? So this much you have studied until your last lecture, and just one instruction, children, that as soon as you join a live class, please do mark your attendance by writing your 5-digit code number and your full name, so that we can mark your attendance in a convenient way, right? So now in today's lecture, we will discuss about another important component of the cell, which are called cell organelles, which are known as cell organelles. So where are these cell organelles present? These are present inside the cytoplasm. So student, in 8th standard, you have learned the difference between cytoplasm and the protoplasm, right? So what is protoplasm? Protoplasm is the complete life-giving substance inside the cell, including the plasma membrane. That complete substance which has got plasma membrane, cytoplasm, nucleus, organelles, that complete collection is known as protoplasm, right? And the transparent jelly-like material which occur between nucleus and plasma membrane, that jelly substance is known as the cytoplasm. So we say that cell organelles. Organelles means tiny organs. Organelles का मतलब होता है बहुत ही ज़्यादा छोटे organs. I hope you know the meaning of the word organs. Organs means you have learned that cells combine to form tissues. Tissues combine to form organs, right? In the similar manner, inside a cell, organs of cells are present, and we call them as cell organelles. We call them as cell organelles, right? So in simple words to define them, that these are tiny membrane-bound structures. Means they are surrounded by a membrane, and in most of the cases, Not in everyone, but in most of the cases, the membrane is double layer, right? So we say that these are tiny means very small. Membrane bound means they have got a membrane around themselves. Structures present in a cell. And where they are present? They are present inside the cytoplasm, right? And students do remember that cell organelles sometimes are also referred as protoplasmic bodies. Another name. They are also known as protoplasmic bodies, or we also call them as organoids. Means another name as protoplasmic bodies or organoids, right? So these are cell organelles, like about which you already got an introduction in its standard. Endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, etc. All these are examples of cell organelles. But before moving on to each individual cell organelle, first question comes that why a cell need these cell organelles? Why a cell need these organelles? So reason is that cell in itself is a complex entity. And if you talk about the cells of multicellular eukaryotic organisms, then they have got a comparatively complex body design. So for the maintenance of such kind of cells and for that is the performance of different functions which a cell is performing in these organisms, these complex organisms, lots of chemical activities are required. So these activities are being performed inside a cell, but to prevent the mixing, to prevent the intermixing of these different kind of chemical activities with each other, a cell has got tiny organs which are called cell organelles. Means one particular component will be releasing energy, another component will be transporting substances, another component will be secreting substances. So these activities should not get mixed with each other. So because of that only, a cell is having cell organelles, right? So to begin with today, first we will take the cell organelle called endoplasmic reticulum. Endoplasmic reticulum, and in short, sometimes we refer it as ER. Short में हम इसको कई बार ER भी लिखते हैं, right? Now endoplasmic reticulum to define. So first we say that it is an electron microscopic cell organelle. Electron microscopic means this cell organelle is only visible clearly under an electron microscope. Means you won't be able to see endoplasmic reticulum under an ordinary light microscope which we generally uses in our laboratories. Okay, so to see ER, endoplasmic reticulum, we require that are electron microscopes, right? Another thing is that this endoplasmic reticulum, it is a kind of network. It is a kind of network, and this network is made up of membranous structures, and these membranous structures are cisternae, vesicles, and tubules. Means in short, you can say that endoplasmic reticulum, it is made up of three components: cisternae, vesicles, and tubules, right? Now, if you have a look over cisternae, then you can see that these are membrane-bound tube-like structures, and their membrane is studded with ribosomes. Yes, over the membrane of cisternae are present ribosomes, right? Then comes the vesicles. So what are vesicles? Vesicles are basically, you can say, oblong bags-like structures. Oblong means which are not properly spherical. Oblong word का मतलब होता है जो properly round shape नहीं है. Okay? So these are oblong bags-like structure, and generally they contain that is secretory substances filled inside them. And then comes finally that are tubules. Okay, so what are tubules? Tubules are generally branched structures which are narrow tubes. बहुत छोटी-छोटी narrow tubes होती हैं वो generally branched होती है, right? So these three cisternae, vesicles and tubules together, they form endoplasmic reticulum inside the cell, right? So if it is asked in examination, describe the structure of endoplasmic reticulum, then you have to write about this particular endoplasmic reticulum that is made up of three components: cisternae, vesicles and tubules. Okay, so any idea from your side that who discovered endoplasmic reticulum? Who discovered endoplasmic reticulum? Yes? Who discovered endoplasmic reticulum? Any idea? Okay, so I got the first answer from Hardik Jain. Very good. And that is, you are correct only. Porter and Thompson independently, they discovered first endoplasmic reticulum. Yes. दोनों ने अलग अलग अपने हिसाब से इसको डिस्कवर किया था. But in most of the books, only name Porter is given, Keith Porter. The complete name was Keith Porter, but it was discovered by independently by two scientists, that is Thompson and the Porter. Very good, Hardik. Okay. Now let's see about types of endoplasmic reticulum. Okay. So now types is, that is SER, and another one is RER. Yes. So SER. SER means S stands for smooth. And ER we already know, endoplasmic reticulum. So complete name for SER goes smooth endoplasmic reticulum, right? Similarly here R, it stands for rough endoplasmic reticulum. So this categorization of types of endoplasmic reticulum is done basically on the basis of the appearance of endoplasmic reticulum under a microscope. So in case of SCR, they appear smooth. Why? Because they don't have any ribosomes attached to their surface. So when we observe SCR under microscope, we don't see any kind of ribosomes or granular appearance over the membrane of SCR. So we call them as smooth endoplasmic reticulum. Here in RER, the appearance is rough, or we also say that it is granular because it is having attached ribosomes. So RER appears rough under a microscope. Why? Because it has got attached ribosomes. Or it also appears to say that it appears as granular. तो सरफेस पे हमें इसके से डॉट डॉट जैसे स्ट्रक्चर्स दिखते हैं माइक्रोस्कोप के नीचे. तो क्या है वो डॉट्स? These are the ribosomes, right? So that are rough endoplasmic reticulum. Another is that you will always found SER near plasma membrane. A smooth endoplasmic reticulum is always found near plasma membrane. You can see in the diagram that I have drawn here near the plasma membrane. तो सेल के अंदर जो SER है वो हमेशा आपको सेल मेम्ब्रेन के पास मिलेगा, राइट? Whereas rough endoplasmic reticulum, it is always observed near nucleus. In fact, it is believed that rough endoplasmic reticulum, it is formed from the outer membrane of the nucleus. That's why in most of the diagrams of a cell, you might have noticed in various books that endoplasmic reticulum, that is rough endoplasmic reticulum, is shown in attachment with the outer nuclear membrane, right? So that is based upon the location. And as I told you right now, that SER, it is formed from RER only. How SER is formed? When rough endoplasmic reticulum, it loses ribosomes, it becomes, it becomes SER. Okay? So here that is minus ribosomes, you can say. So RER minus ribosomes equals to SER. Here, rough endoplasmic reticulum, this we get from the nucleus. RER किससे बनता है? Nucleus की membranes, right? Another is smooth endoplasmic reticulum. They basically synthesize lipids. They basically synthesize lipids. And about lipids, you might have got an introduction while studying the structure of plasma membrane. That lipids are complex kind of fat molecules, right? Rough endoplasmic reticulum, they synthesize proteins. They synthesize proteins. Now, white rough, they contain ribosomes, and the main function of ribosomes is to synthesize proteins. So, main function of RER is to synthesize proteins, right? Another one is that SER. It performs one important job, and that is in the liver cells of the vertebrates. What is the function? That it detoxify the poisonous substances. Yes, there are certain poisonous substances which when go to liver, then their poisonous nature is killed or removed by the action of the smooth endoplasmic reticulum, which is present in the cells of the liver. Right? And that's a kind of boon for the people. But such kind of function is not performed by rough endoplasmic reticulum. Right? So, children, just now I told you that SER synthesizes lipids and RER it synthesizes proteins. And you have also discussed that the plasma membrane it is basically made up of lipids and proteins. There you have studied about its trilaminar structure, that 2 layers of lipids and 1 layer of protein, right? So those lipids and proteins which together make up a plasma membrane, these are synthesized from SER and RER, right? So when synthesis of plasma membrane takes place from these lipids and proteins, synthesized by SER, then this process is known as membrane biogenesis. This is known as membrane biogenesis. And yes, it is an important term for examination. So what is membrane biogenesis? That when lipids and proteins synthesized by endoplasmic reticulum are used for the generation of plasma membrane, the process is known as membrane biogenesis, right? Now, that is function. What are the functions of endoplasmic reticulum? So, first one is that it acts as circulatory system of cell. Circulatory system of cell. In 7th standard, you learned about your own circulatory system that is comprised of blood vessels and heart. And the main job of circulatory system is transportation of substances to various parts. In the similar manner, the channels which endoplasmic reticulum is being forming in the whole of the cell. Through these channels, various substances are transported in various directions. Since it is involved in the transportation of substances, so we say that it forms the circulatory system of the cell. So sometimes we get a question in exam that which cell organelle is referred to form the circulatory system of the cell. So that is endoplasmic reticulum, right? Another one is that it provides a kind of cytoplasmic framework. The cytoplasmic framework means, or I should say, cytoplasmic structural framework. Because when we talk about the floor of a cell, that is made up of cytoplasm. and cytoplasm is liquid. Basically, it is gel-like substance. So various kind of activities need to occur inside the cytoplasm. So to provide a kind of structural stability, what happens? This endoplasmic reticulum comes in. तो जो ER की जो आपकी सिस्टम में है, जो उसकी जो सरफेसेस हैं, जो आपकी ट्यूब्यूल्स हैं, उनकी जो सरफेसेस हैं, उन सरफेसेस पे बहुत सारी बायोकेमिकल एक्टिविटीज डे नाइट कंटिन्यूअसली होती रहती. सो इसे दैट it gives a kind of cytoplasmic framework to the cell, means to some extent it is responsible for maintaining the that is so called shape of the cytoplasm of a cell, and it also lend its surface for various kind of biochemical activities, right? Another thing of function you can include from your side itself, the function of SER, the function of RER, that is synthesis of proteins and lipids. Okay, and one more is there which you can remember easily, that SER, it helps in synthesis of ascorbic acid. Yes, it has been found that the smooth endoplasmic reticulum, it helps in the synthesis of ascorbic acid. And children, the functions which endoplasmic reticulum performs, it is a long list. And basically, I am focusing here on those functions which are mentioned in your NCERT. Although I told you one extra also this one, because it is the shortest one you can remember easily. Okay, so in all functions, if it is asked for 3 to 5 marks, you can write easily cell circulatory system, structural framework to the cytoplasm of the cell, biochemical activities. RER synthesizes ribosomes, SER synthesizes lipids, they help in the generation of plasma membrane, right? SER helps in the detoxification of poisons, okay? SER also help in the synthesis of ascorbic acid. So, you have got a good list with you to write the functions of endoplasmic reticulum, right? So, that is about endoplasmic reticulum as a cell organelle. Okay, and one more thing, you have studied about the type of cells based upon the kind of nucleus they are containing, that is eukaryotic and prokaryotic. So do remember that prokaryotic cells, they don't have membrane bound cell organelles. Are you getting my point? So in case of prokaryotic cells, in place of cell organelles, what happens? There are different regions which behave like organelles. A complete cell है, say for example, cell का ये corner endoplasmic reticulum की तरह function कर लेगा. Cell का ये corner mitochondria की तरह function कर लेगा. Like that means membranes are missing. And what is the significance of membranes? That the chemical environment of one organelle get mixed with the chemical environment of the another organelle. So, do remember that membrane-bound cell organelles is one of the prominent feature of eukaryotic cells, and it is not there in prokaryotic cells, right? So, that is about endoplasmic reticulum. Now, let's take another one, and that is, yes, okay. So that one is, yes, next one is Golgi body. Yes, Golgi body, Golgi complex, or it is also known as by the name mitochondria, or one more name, idiosome, or one more is there, that are called Dalton's complex. Yes, these are the different names for Golgi complex, right? Golgi body, which we say, and Golgi body was discovered by the scientist called Golgi. And do you remember these were discovered in the nerve cells of cat, right? So sometimes it is asked in your competitive examination, name the cell organelle which were discovered inside the nerve cells of the cat. And the person who has discovered, that is Camilo Golgi. So to honor the person for this discovery, this cell organelle was named after him only, that is Golgi body. And another names goes mitochondria, idiosome or Dalton's complex, right? Now structure wise, like this we see. Okay, so that is the outline figure diagram for a Golgi complex. Remaining structure is very detailed, पर डायग्राम में थोड़ा सा ध्यान से देख के रखेंगे. अगर ऐसा कोई फिगर आपको कभी एग्जाम में मिलता है आइडेंटिफाई करने के लिए, then this diagram belongs to Golgi body, right? And with reference to the structure, it is also composed of, that is cisternae, vesicles and vacuoles. Yes, in place of tubules, here we have got vacuoles, right? And like endoplasmic reticulum, the structure of the membrane of Golgi complex is also similar to that of plasma membrane. Means the membranes of Golgi complex are also made up of 2 layers of lipids and 1 layer of protein. Means they are lipoproteinaceous in nature. Okay, so that is remaining regarding the structural components. So do remember that is cisternae, vesicles and vacuoles. In place of tubules, Golgi complex they contain, that is vacuoles, right? Now this, that is Golgi complex. If we talk about functions, so first is, that is, it is known as the packaging house of cell. It is known as packaging house of cell. Why? Because it is involved in the storage, modification and packaging of various cell secretions into vesicles, right? तो जो भी सेल के अंदर सीक्रेशंस होते हैं वो गोल्जी apparatus के पास आते हैं. तो जो गोल्जी apparatus है उनको, फॉर एग्जांपल बहुत सारी डिफरेंट टाइप की प्रोटीन्स होती हैं, राइट? एंड दीज प्रोटीन्स दे हैव गॉट अ वेरी वाइड एंड वेरी स्प्रेडफुल स्ट्रक्चर. So for means of transportation, what Golgi complex do? It compresses the proteins, right, and pack in a convenient manner inside these vesicles, and these are transported from one place to another. तो उसके स्ट्रक्चर को हल्का सा मॉडिफाई करके कन्वीनियंट वे में वो पैक कर देता है वेसिकल्स के अंदर, जिसकी वजह से ट्रांसपोर्टेशन आसान हो जाता है, राइट? दैट्स व्हाई वी कॉल इट एज the packaging house of the cell. What happens inside the packaging house? You just give any material to them and they will do it's such a packaging that you can transport it easily from one place to another. Similarly, the same job is performed by the Golgi body inside a cell. It is known as the packaging house of the cell, right? Another one is that it helps in the synthesis of complex sugars. Yes, complex sugars which we commonly call them as polysaccharides, right? So various kinds of polysaccharides are synthesized by different kind of sugars like monosaccharides, disaccharides. तो बहुत सारे सिंपल शुगर्स से मिलकर कॉम्प्लेक्स शुगर्स का सिंथेसिस होता है और वो सिंथेसिस भी कुछ एक्सटेंट तक Golgi complex के अंदर होता है, right? So we say that synthesis of complex sugars from the simple sugars. Another one is that is formation of, that is lysosomes, a bit part, right? So some part of lysosomes, it is being formed by this Golgi complex, right? So generally from Golgi complex, they will make you get the question from the term packaging house of the cell. Generally, the question is asked for 1 mark. What is the main function of the Golgi complex inside the cell? Storage, modification, and packaging of the substances which are secreted inside the cell for easy transportation. Right? That is about Golgi complex. Okay. Now, next one is the next cell organelle in the sequence. Yes, that are lysosomes. That are lysosomes. So, with reference to draw the diagram, so that is membrane, and it is known as matrix. Okay. Now, what are lysosomes? We say that these are the small vesicles, right? And these vesicles, they are filled with strong digestive enzymes. Strong digestive enzymes, right? So membrane bound vesicles which are filled with strong digestive enzymes are called lysosomes. And do remember that lysosomes are also electron microscopic by nature. Means that they are visible under only an electron microscope. Okay. Now, structure-wise, it is having a membrane. Okay, and inside the membrane is present the substance, the floor of the substance, which is in the floor of the cell or this vesicle is called a matrix, right? And students, this matrix consists of almost 40 different kind of enzymes. Yes. Inside this small lysosome, in the matrix are present 40 different kind of digesting enzymes. 40 different तरीके के enzymes present होते हैं lysosomes के matrix के अंदर. Right? Now the functions of lysosomes means which are very important, and to remember functions easily, so you just remember the term. So the first one is called heterophagy, second is called autophagy, and the third one is called autolysis. To remember in short the functions of lysosomes. First one is called heterophagy. Hetero stands for others and phagy means to digest. Hetero word का मतलब होता है दूसरों का, others, और phagy का मतलब है to do the digestion. So what happens? The one important function of lysosomes is that to digest the foreign material. To digest the foreign material means if there is any bacteria which is entering the cell. So these are the lysosomes which are going to digest that particular bacteria because of by using their strong digestive enzyme. Similarly, if there is any kind of food is entering the cell, then again it will be digested by the strong enzymes which are present inside the lysosomes. So it is known as heterophagy, means when lysosomes enzymes they digest any foreign material which is entering into the cell, means which is not a regular part of the cell, but it is entering inside the cell, When such kind of material is digested by the lysosomes, the phenomenon is known as heterophagy, right? Another one is autophagy. Now what happens that inside cell many kind of cell organelles are present, and due to the continuous functioning of the cell organelles, what happens that these cell organelles eventually becomes older. Okay, and some or that you can say more or less parts of there they start disintegrating. So what happens? These old and worn out cell organelles, they are present inside the cell only. So to keep the cells clean, what happens is there is any dead cell organelle, there is any dead remain of any old cell organelle, then all kind of waste which is generated inside the cell like this, these are digested by the lysosomes, and this is known as autophagy. Auto stands for cell, means जिस सेल में लाइसोसोम्स प्रेजेंट है उस सेल के अंदर जो भी वेस्ट है, जो डेब्रिस है, उसको डाइजेस्ट करके क्लीन करना, दैट इज द मीनिंग ऑफ द वर्ड ऑटोफैगी, राइट? दैट इज नोन एज ऑटोफैगी. ओके, सो दैट इज इट डाइजेस्ट द वेस्ट इन सेल, राइट? Okay, and that's why it is also referred as that lysosomes, they make the waste disposal system of cell. Yes, the cellular component, the cell organelle which is responsible for building up the waste disposal system of the cell is lysosome. Or question can be asked, name the cell organelle which is responsible for keeping the cell clean. That is again lysosomes, right? And another one is autolysis, and also called as autolysis. Autolysis. Okay. Now auto stands for cell, and lysis means to break down, or you can say destruction, means to cause self-destruction is known as autolysis. So what happens, these lysosomes are quite strange organelles. What happens, whenever there is an injury in the cell, or whenever there is a disturbance in the chemical nature of the cell, then what happens, these lysosomes, they rupture and they release enzymes. So for example, किसी सेल में इंजरी हो गई, ठीक है, तो जैसे सेल के अंदर इंजरी होगी उसके अंदर का केमिकल एनवायरनमेंट डिस्टर्ब हो जाता है. So because of that, what the lysosomes do, they rupture. वो अपने आप को रप्चर करके पूरे सेल के अंदर जो उनके अंदर स्ट्रॉन्ग डाइजेस्टिव एंजाइम्स हैं, 40 डिफरेंट टाइप्स के सारे एंजाइम्स को सेल के अंदर रिलीज कर देते हैं. And gradually what happens, that these enzymes digest the whole cell. Yes, it digests the whole cell. Now since the cell inside which lysosomes were surviving, it is no more, and because of that lysosomes, they also came to end. वो खुद भी खत्म हो जाते हैं, अपने सेल को खत्म कर लेते हैं और फाइनली वो खुद भी खत्म हो जाते हैं. So they are causing cell destruction. That's why we call them as suicide bags of cell. Suicide bags of the cell. And that's a very, very important term which you have to remember from this topic of lysosomes, because every year one question is for sure going to be asked from this particular common term, that what are suicidal bags, or which cell organelle is known as suicidal bag of the cell. Or they ask you the direct question, why lysosomes are known as the suicide bags of the cell. So reason I told you that whenever there is an injury or accident occurs with the cell, then what happens? Lysosomes, they rupture out, they release the enzymes into the cell. These enzymes digest the complete cell, and finally the lysosomes also, that is, vanish. So since they are digesting they are causing self-destruction. So we call them as suicidal bags of the cell. Right? Okay. And do remember it. Again, I am telling, बहुत important है बेटा, ये हमसे question हर साल एक बार जरूर आता है. That why lysosomes are referred as the suicide bags of the cell. Okay. So these are the three important terms which can be referred to as the functions of lysosomes. Not only this, lysosomes, they also release their enzymes out of the cell for digestion purpose, right? So these enzymes are packed in the vacuoles, and by the process of exocytosis, these vacuoles are released out of the cell. तो ऐसा नहीं कि जो लाइसोसोम्स है वो सिर्फ सेल के अंदर का डाइजेशन करता है, नो, इट आल्सो कैरी आउट एक्स्ट्रा सेलुलर डाइजेशन, राइट? सो रिगार्डिंग द फंक्शंस ऑफ लाइसोसोम्स, आई टोल्ड यू, heterophagy, they digest the foreign material; autophagy, they digest the waste which is generated inside the cell; and autolysis, they sometimes under special conditions, situations, they digest their own cells. Because in textbook you may not find these terms, but these are important for your competitive part. So do remember what is heterophagy, what is autophagy, and what is autolysis. Okay. So student, that's all about for this particular class. So what you have to do, whatever we have discussed today, especially I discussed with you about 3 cell organelles: endoplasmic reticulum, Golgi body, and lysosome. So you have to read this much portion from your NCERT. Okay, and if there are, if any doubts are there, then you can put a line to my WhatsApp number also. अगर आपको लगता है बाद में मैंने NCERT पढ़ी और अब मुझे समझ में नहीं आ रहा, then feel free to drop a line to me. I will be very happy to see your doubts. Okay, so I hope you enjoyed this session with me. We will meet soon again in next more session. Till then, take care, stay safe, happy learning. Bye.